Good afternoon, students. My name is T.THIENPRASERT. I'm your lecturer for today. In this class, we are going to study Laboratory Chapter 2, entitled Spectrophotometric Analysis of Color Solution. In this chapter, we aim to study basic principles and also analyze spectrum and absorbance of color solution by using spectrophotometry. Let's start with a brief introduction on light spectrum. Light spectrum is the range of electromagnetic spectrum from 200 to 700 nanometer. It can be divided into spectral ranges, which are ultraviolet or UV light from 200 to 400 nanometer, and the visible light from 400 to 700 nanometer. As can you see from this figure, the absorption of light occurs when the appropriate energy of photons or from the light is absorbed by a sample and excites a valence electron from the ground state into the excited state. The light absorption is considered as the unique property of sample because each sample has a unique number of electrons that exist in different atomic orbitals. Therefore, each sample absorbs light differently. The color of the solution that we see is due to the reflection and transmission of light from the absorbed molecules to our eyes. For example, riboflavin, or we know as vitamin B2, is yellow because molecules of riboflavin absorb red and blue light and transmit yellow light into our eyes. Today, we are going to use riboflavin solution. Do you know where we find it in nature? Riboflavin can be found in grains, plants, and dairy products. It's dissolved in water and is stable to heat, acid, and oxidative reaction. However, it is sensitive to light. It functions as cofactor and it is crucial for breaking down food components, absorbing other nutrients, and maintaining tissue. It's also important for normal growth. Here is riboflavin structure. Riboflavin consists of 3 aromatic rings. Thus, it has absorption spectrum in both UV light and visible light. Now, what is a spectrophotometry? It is a technique used to analyze physical properties of biomolecules in both qualitative and quantitative measurement based on the amount of light that absorbs by sample. It is used for analyzing absorption spectrum and concentration of solution because it is simple, quick, and accurate. Moreover, it can also be used for micro-volume samples and sample mixtures. In order to obtain the highest sensitivity and the most accuracy, measurement of sample must be performed with a wavelength that gives the greatest absorbance. This is known as lambda max, or it is also known as lambda submax. The value of lambda max has a unit of nanometer, and it is a characteristic value of each sample depending on the condition of experiment. For example, both DNA and protein have absorption spectra in the UV light, but they have different value of lambda max. As you can see, lambda max of double-stranded DNA or single-stranded DNA are at 260 nanometer. This due to the absorption of nitrogenous base, while proteins have the lambda max at 280 nanometer because of the appearance of aromatic rings from tryptophan and tyrosine. Now let's see how to measure absorbance of sample from this figure when the monochromic light pass through a solution. From beginning, the abbreviation using as I0. Once it passes through the solution, fraction of light is absorbed by molecule in the solution. The transmitted light therefore has lesser intensity, which used as I. The amount of light that is absorbed by a solute can be measured in terms of absorbance, which represented by A. Or it is called optical density or OD, as indicated by this equation, when A absorbance equal log of I0 over I. For spectrophotometry, there is one main law known as Beer-Lambert law, which consists of the Beer laws and the Lambert law. For the Beer law, A equals Kc, meaning that the absorbance of light is directly proportional to the concentration of the solution. On the other hand, the Lambert law, A prime L. This means that the absorption of light is directly proportional to the path length of the light passing through the solution. So together, the Beer-Lambert law indicates that the absorbance of light is directly proportional to the path length of the sample and the concentration of the solution. The equation of Beer law is A equal epsilon Cl. This funny E is the fifth letter of Greek alphabet called epsilon. It is single experimental constant of the Beer-Lambert law. We call it a molar extinction coefficient or molar absorbance coefficient. It is a unique value for a particular sample depending on the wavelength and the experimental condition, such as temperature, solvent, and pH. It has a unit as 1 over meter and centimeter. Please note that if concentration of solution is presented in gram per liter or milligram per milliliter, we cannot use molar extinction coefficient anymore. In contrast, a constant of Beer-Lambert laws in this case is absorbency index or an extinction coefficient or capital E. Please note that there are three assumptions in Beer-Lambert law, including first, the radiation is perfectly monochromatic. That means it has a single wavelength. Second, there are no molecular interaction between the absorber and other molecules in solution. And lastly, the solution has to be homogeneous. Importantly, the absorbance of light that obeys Beer-Lambert law is displayed as a linear regression curve passing through the origin, meaning zero for all two dimensions. However, the linearity of the Beer-Lambert law is limited by the high concentration of acidic bases and salted solution, including some organic solutes that tend to coagulate together. These high concentrated solutions generate deviation from linearity, which are positive deviation and negative deviation. From this figure, solution 1 indicate a straight line at a lower concentration. Notably, the line passed through the origin in this case. Line number 2 and line number 3 represent limitation of Beer-Lambert law at a higher concentration of solution, which result in a positive deviation and a negative deviation respectively. Next, we will talk about spectrophotometer, which is the device that's used to quantify the amount of light transmitted through solution. The measurement is analyzed in terms of percentage of transmittance from an equation I over I0 times 100. Therefore, it is a linear scale from 0 to 100%. However, a term of absorbance, bigger A, is commonly used in practice. Absorbance can be calculated from 2 minus a logarithm of percentage of transmittance. Therefore, the absorbance is a log scale from 0 to 2, and it can be read accurately in the range of 0.3 to 0.6 for an analog spectrophotometer. Therefore, the concentrated solution should be diluted before the test for the accurate reading. Major advantage of spectrophotometric technique in biochemistry includes identification of sample, analysis of concentration. This can be done by using Beer-Lambert law and by comparing a standard curve. Moreover, it can be used to monitor the reaction progress. For example, a spectrophotometer can be used to monitor the reaction of NAD+, right here, to NADH by lactase dehydrogenase or LDH, which is the enzymes. From figure, both NAD+ in the oxidized form and NADH in reduced form can absorb light at the wavelength at 260 nanometer. However, only NADH can absorb light at a wavelength of 340 nanometer. Therefore, we can determine the direction and quantitative rate of reaction by monitoring absorbance of NADH at a wavelength at 340 nanometer. For example, if the reaction favors the reduction of NAD+ to NADH, then the absorbance will be increased over time. In contrast, if NADH is oxidized to NAD+, the absorbance value at this wavelength will decrease over time. So thank you very much for your attention.